happens. Ooh, Wednesday.
Okay, good. Okay.
And how do I sign in?
What's that?
I need to sign in as well.
Yeah, okay.
And how do I do that?
That, well, you'll be using this, right?
Yeah.
So I guess we ought to cut the video off.
Oh, wait, I'm on here. Right.
Oh, yeah. Okay. Why don't we stop video until we're ready to
Okay.
go up here? So we're not here. Okay. Careful.
So I can just be you, perhaps.
What's that?
Yes.
And if I can't sign in because I.
Yeah,
can't
yeah.
sign in.
I'm assuming we're projecting from here. Let's do that.
Now,
Turn
are
the
we
video
muted?
back.
Yes, turn the video back on.
Leave mute off. And
Turn that video back on. Hey, guys.
well, how come
Hello.
we're not showing anything on here on the screen. Turn it back on mute. Oh, you got it off. Right. No.
Okay, there's that button on the wall that's supposed to allow you to start mirroring what's on your computer. Remember, it's to the left of where you're standing. How's it going, Carson? There's Alden.
My wise president, keep doing the good job.
Can you hear us okay, Carolyn? Okay.
But we can't hear her.
That's
Yes,
okay.
I am unmuted now.
Okay. All right. Good.
Good, good.
All right.
Did y'all get it to display on the screen behind you?
Sure.
Further back.
Good, good. That's all we needed to know.
So just go down to this corner, just find the setting, duplicate or extend.
Until where you can find your
And then
presentation.
the setting right here is on duplicate, just hit extend these displays.
Okay.
Yeah. And then once you pull up your presentation, it'll automatically give you the two different views on the screens.
Perfect.
You might have to hang around the front, Alden.
Yeah. I mean, I'll be right there.
that's
So,
Okay.
Yeah. So, okay. So how do we get us back on here?
sorry. If you want to pin your video, just click these dots.
Oh, okay. Okay.
Ah.
Good. Okay.
Cool.
Yeah, great. So
Okay.
we'll just leave it blank till
All
we're ready to
right.
start here in a few minutes.
Okay.
Then we'll
Awesome.
have, it's unmuted.
Hi, how are you?
Good, man.
Good, good to see you.
Yeah, oh, yeah, yeah. So there, this, everybody. Yeah, they got coastal area,
Yeah, I've seen that.
Yeah, yeah. Like I said, they got. If I could get everyone to come in, we're ready to start the program, please. Get my best side, Jay, which is the back. I want to open up the meeting. Can you guys hear me without the microphone? Okay. All right. My name is Carson Stokes. I'm the Vice President of the Heartwood chapter of Texas Master Naturalists. I want to welcome everyone here tonight that's at the Timberlane Community Center and all my Zoomers out there that are, can you hear me okay? Yeah. Shake your head out there. Okay, good deal. I just wanted to do a little bit of business related to Heartwood, and then we'll get the program going. There's a lot of exciting discussions going to be made. This is officially called the Student Research and Education Symposium. Symposium. These are interns that work for various parts of nature. I mean, of Mercer and Harris County. I think they have some really interesting presentations to make tonight. But just a few logistical things. Just a reminder of future meetings. September, we're going to have a presentation by Wizzy Brown on ants. We couldn't get E.O. Wilson, so we had to go with Wizzy. Okay. In October, Claire Moore is going to discuss birding programs, opportunities to learn more about birding, et cetera. And in November, we're hoping to have, it'll be our last meeting of the year. We're hoping, depending on what goes on with COVID, to have a party. So we'll see. Yeah, we'll see what happens with that. Kind of, recognize that people have done an outstanding job as volunteers. It's been a tough year for all of us. Hopefully it won't be as bad as last year. But anyway, so with that, let me go ahead and, let's see. I'm sorry. Anita, I think will you come up and announce the people that are involved with Mercer, or do you want me to do that?
Oh,
Okay, we're ready. That's
on
our,
a separate section of the agenda.
yeah.
All right, you're starting early.
Now, I'm going to start at 5:45.
Oh, okay. Okay,
So,
cool.
so this should take
All
me
right.
15 minutes.
So you didn't need the 15 minutes?
No, I did
Okay.
not. So you can have it. I'll give it to you.
Then that would be
for free.
our right honorable Chris Ludwig.
Want to welcome y'all tonight. So y'all can kind of see what our interns worked on over the summer at Mercer. I want to thank them for all the hard work they did at the gardens and at NBC. Kind of getting some of the things taken care of we needed. I know the NBC interns worked with Linda going through the inventory and stuff like that. So that was a big help, I know. And then with y'all in the gardens, doing a lot, getting us, you know, ahead of the game for when fall comes and stuff like that. So look forward to seeing y'all's presentations and I hope everybody enjoys. Thank y'all. What's that? There. That better?
Awesome. Well, hello, everybody. We're starting a little bit earlier, and I am so glad we're here in person this year. I can see some of you through the plants, so welcome over on that side. Thanks, Chris. Again, welcome, everybody. Thanks so much to Carson for being here and introducing us. And as always, thanks to our Precinct 4 Commissioner Jack Cagle and our Parks Administrator, Dennis Johnson, all our P4 staff, Precinct 4 staff, our volunteers, everyone for making this symposium possible. Special thanks to Bud Gessel with the Timber Lane Utility District for making this beautiful community center available to us tonight. So I hope everybody's enjoying that, the great displays around this community center. Thanks always to Heartwood chapter of the Texas Master Naturalists for co-hosting this annual event since 2015. Heartwood volunteers have been a part of Mercer's volunteer family since 2005. Even during COVID lockdowns, our volunteers have provided amazing off-site support to Mercer. We thank our interns, professors, their families and friends for nurturing and supporting these fine students. And what excellent work the interns have accomplished this summer. We thank, of course, Precinct 4 for funding 4 of the 6 interns presenting this evening. Thanks to Mercer's nonprofit, the Mercer Society, TMS, for administering our grants for our 2 Mercer Botanical Center MBC interns. Both Alex and Alden were funded by the Gibson family, as well as the Native Plant Society of Texas, Houston chapter, and the Knowles family in honor of Dr. Larry Brown. Do we have any TMS board members here tonight? I don't think so. Anyway, they're wonderful support to us throughout the year. As many of you know, the Spring Branch Science Center, or the SBSC Botanical Reference Collections, were transferred to Mercer Botanic Gardens as a long-term loan. And you'll be hearing more about our herbarium collections tonight. Our volunteer and P4 staff physically moved the collections to the Botanical Center during the winter of 2016 and the spring of 2017. Included are botanical reference collections of over 1,000 books, 2,300 botanical illustrations, and over 50,000 herbarium specimens. The herbarium collections support Mercer's own core and growing herbarium collections, referred to within the Herbarium World Database as MERCA or MERCA. You'll be learning more about MERCA tonight. And what is a herbarium? Who doesn't know what a herbarium is? Most of you do, but I'll just remind you. A herbarium can be compared to a library of carefully prepared, dried, labeled plant specimens mounted on archival paper and filed alphabetically by the scientific name of the plant. These specimens serve as a reference for botanists, citizen scientists, artists, and more here locally and internationally. With the support from our volunteers and interns, all of the mounted 40,000 spring branch specimens have been quick databased to species and collection location, and many have been fully databased. Our SBSC and MERCA herbarium collections are part of the Texas-Oklahoma Regional Consortium of Herbaria, or TORCH. And in the next years, these specimens will be imaged as part of a National Science Foundation grant award to TORCH. And once imaged, these specimens will be available for online study. Mercer would not have become the recipient of these collections without the recommendation by the Spring Branch Herbarium's primary collector, curator, plant taxonomist, the late Dr. Larry Brown. We miss him very much. And please join me tonight in thanking Dr. Brown's family for continuing their amazing support to Mercer Botanic Gardens. Now, Mercer's Botanical Gardens represent an amazing diversity of plants. Maintained at the MBC, or the Mercer Botanical Center, along with the herbarium database, is the Gardens Living Collections Database. The Living Collections Database is shared with partners at Botanic Gardens Conservation International and the Global Genome Initiative Gardens programs. Thus, our living collections serve as a source of research material to national and international efforts. And just this week, Jacob Martin, our greenhouse manager, and interns prepared a sample of Mercer's young Titan arum, or corpse flower, a native of Sumatra, that we then FedExed to Chicago Botanic Gardens, where they are creating a pedigree for all Titan arums in cultivation to guide future breeding programs for that species. So on with our programs. Jacob will introduce our first speakers, our horticulture interns.
All right, I'm Jacob Martin. I am the greenhouse manager here at Mercer Botanic Garden. I supervised Madeline, Emily, and Rob this summer. They worked in pretty much every garden we had at Mercer. They pulled tons of weeds and they hardly ever complained about it. They learned their heat tolerance limits, and they learned about lots and lots of other plants we have in the garden, and they're going to teach you all about a few tonight. They were fantastic interns, and I hope you all appreciate their presentations. We're going to start off with Emily Flores tonight.
Good evening. My name is Emily Flores, and this is my presentation on my closed aquatic native terrarium. So a little bit about me. I'm currently a senior at Spring High School, and I want to major in architectural engineering. This summer I had an internship at Mercer Botanical Garden. And I took this internship because I wanted to see if I had a passion or an interest in plants and biology. So going further into my internship this summer, I planted new color beds around the Mercer Botanic Garden, specifically in Renaissance and Shakespeare. And we weeded beds and made them look new again. For example, like the Under the Sea garden and the Children's Garden. We relocated fish from the front pond to Story Lake. And my daily task consisted of weeding, watering, making cuttings, and working in assigned areas, which mine was tropical. Here on the right, we can see there's pictures that I took during my internship. Here are just some more pictures. One of them is the bed in the ramble, which is a section in the part of the back of the garden. And here's one of the fish that we relocated. We named it Soul because of its vibrant orange colors. And here's just another bed that we weeded and we gutted and we made it into a new bed again. So going further into my project, I had an interest in aquatic science, and I had an interest in all the ponds in the garden. And I wanted to see if I could replicate the life in the pond inside of a smaller size, which is my jar. So the first thing that I did to creating this jar was gathering sediment, which was the sand in the top layer of the endangered species pond. And I placed it on the bottom of the jar. And then I gathered a bunch of plants. In this case, I gathered the Bacopa caroliniana and I rooted it in the bottom of the jar. And then I finally filled the jar with water from the endangered species pond. So going into the initial contents of the jar, as you can see, there is no initial signs of life except for the plant that I have placed in there. It's mostly foggy because all the sediment was still floating around and you couldn't really see anything but the sand and the undefined Bacopa caroliniana. So here are second day pictures of the experiment. This was taken on June 22nd. And as you can see, now we can see the Bacopa very clearly, but everything still hasn't settled. There's still sediment floating around. And you can notice that the Bacopa still hasn't fully rooted inside the jar, so it was still floating around. To go further into the sediment of the jar, the first layer, which is the top brown layer, is pond sludge, which is a buildup of organic materials such as animal waste, leaves, grass, dead algae, runoff fertilizer, grease, and oil from runoff. And then the bottom layer is sand. And if you notice that there is tunnels running along the jar. And these pictures were taken July 28th. Here's just some more pictures of the sediment of the jar. And you can notice that there is tunnels running along the sediment, and I'll further later explain why there's those tunnels. So throughout my observations, there was many organisms. And here's just a couple of my favorite pictures that I took of them. My personal favorite was when I got a snail and I looked at it under a stereoscope and I took a picture, which is the picture you see in the middle. And going into the organisms, my first one is the Bacopa caroliniana. This picture was taken July 12th. And this is a perennial creeping herb, and it will grow above water if it's able to. However, there's no specific time of year where it goes dormant. And the temperature tolerance is 39 to 84 degrees Fahrenheit. And this plant usually grows in shallow water, bogs, and semi-submerged areas. And it doesn't grow in areas more than 3 feet of depth. In this picture, you can see that from the stem, roots were starting to come out. And I thought that was really interesting because it was starting to latch down into the jar. So here's just some more pictures of the Bacopa. It's part of the Plantagenaceae family. And there is debate whether it belongs to the Scrophulariaceae family, but most of the resources that were up to date suggested that it was part of the Plantagenaceae family. And it's native throughout eastern United States, ranging from Texas to Maryland. So my next organism is the damselfly. These pond damsels usually grow around 1 to 2 inches in length, and their nymphs are found in submerged vegetation and rocks, which in this case, it like to be around the Bacopa and it would hide under the rocks. And it's usually found in environments with still water, and in this case, my jar was an ideal environment for it to grow. So here's just another detailed picture of the damselfly. And it's part of the Coeninagrinae family, and it's native to the eastern United States and Canada. And its temperature tolerance is around 68 to 87 degrees Fahrenheit. So my third organism is the sludge worm, and it inhabits cohesive muds. And in this case, it would love to make tunnels and inhabit the putch lawn that I had placed in the jar, which is what these pictures are. As you can see, the sludge worm is digging deeper into the sediment, and I noticed these tunnels around July 28th, which was 5 weeks into my experiment of the jar. It was really cool to see it just create all these little tunnels and make all these little patterns along the jar. And here are just a few more organisms that I found in my jar. There was an Asian tiger mosquito. I did see how it grew from a larva to a full-grown mosquito. Unfortunately, I don't have those pictures, but it was really cool to see it release. And then there was this amphiphod, which was a shrimp-like looking organism, and there was many snails and water fleas. So here are some progress pictures of my experiment. My focal point is that there was a lot of algae growing in the jar, which suggests that there was a lot of exposure to light. These pictures were taken around July 15th. So here's what I learned from my experiment. In this experiment, I wanted to compare the life of the pond to the life of the jar. So when it came to temperature varied around 85 to 95 degrees Fahrenheit throughout the summer, while the temperature consistently stayed at 73 degrees, and the jar stayed in the potting shed. And when it came to shade, the pond had morning shade, but it was mostly exposed to full sun with the shaded cover of trees. And when it came to the jar, it stayed in the potting shed, which has a 60% shade cloth on top of the building. When it came to animals, there were larger animals, which were mosquito fish, turtles, frogs. And in my jar, there was smaller animals like bladder snails, amphipods, and microscopic animals. When it came to oxygen, it had a constant flow. The pond had a constant flow of oxygen. However, the jar, there would be weeks where I didn't open the jar. However, I suggest that the Bacopa was the main source of oxygen for all the organisms inside the jar. So to go furthermore into the pond, Unfortunately, many of the Bacopa were disappearing as time progressed. This picture was taken around July 15th. You can notice that there's only a few Bacopa left, and here are the reasons why I suggest that the Bacopa was disappearing in the pond. It could be because the temperature was inconsistent and the heat went over the tolerance of the Bacopa. As you recall, I mentioned that the heat tolerance was 84 degrees, and this summer was a very hot summer. So it was around 95 to 97 degrees. And I think that upset the Bacopa. There was also larger animals feeding on the plants, such as turtles. In my jar, there was only a little bit of animals feeding on the Bacopa, but they would never feed on the stem. They mainly feed on the roots, while as in the pond, they would feed on the stems, leaves, and they would disrupt the whole Bacopa. And there was also flooding because of the rain, and flooding can also take fertilizer from beds into the pond, which it's really bad for aquatic life and aquatic plants. And there was also artificial bacteria added, which could have also upset the Bacopa. So here's what I learned throughout my internship. I learned how to make cuttings, which is you take a part of a plant and then you place it in a little soil with growth hormone and it grows into a full-grown plant. And I learned my pain tolerance to different insect stings. As you can see on the left, I got stung by an asp, which if some of you don't know is a hairy caterpillar. I don't recommend to touch it. There is, on the right is when I got stung by a wasp the first time. And, you know, I got stung by many mosquitoes, ants, but those are just the main ones that I wanted to point out. And I learned about the different types of weeds, how they grow and how to take them out and how to get rid of them. Here are just a few pictures that I took throughout my internship of the beautiful lilies in the front pond. And one of the pineapples that my boss Jacob had grown. And here's just a few more flowers that I found really pretty and a succulent. And here's just some of the animals that I encountered. As you can see on the right, there is a little frog hiding in the pine straw and a nice tree frog that I released. Here are my sources, and I just want to thank Mercer for giving me this opportunity to have this internship, and thank you.
Questions for Emily? That was wonderful, Emily. And now Maddie, you're up next.
Do I need to do the duplicate thing?
It's like technical.
Do you have it on the PowerPoint?
Actually, no, it's on a PDF.
I think you can do it this actually. Okay, that's a little bit better.
Thank you. Good evening, ladies and gentlemen. My name is Madeline Rodriguez, and tonight I will be presenting on various plants at Mercer Botanical Gardens that have medicinal properties. Before we get into that, I would like to tell you a little bit about myself, as well as about my hopes and plans in the future, and a little bit about my summer internship experience at Mercer Botanical. I'm currently a student at the University of Dallas. I am going into my senior year. I'll be graduating in the spring of 2022 with a BS in biology. I hope to attend UTSA grad school and pursue an MS in geology and specialize in paleontology. The specific branch is still up for grabs, but paleobotany has been a contender since high school. I found this internship through my brother, who is a photographer and frequently shoots at Mercer. And so I've walked the grounds many times and seen the multitude of beautiful plants. Additionally, I wanted to have an internship prior to graduating to get some experience in a biological field. And since I had interest in botany prior to exiting high school, this was going to show me why I hadn't pursued it or prove me wrong and spark interest in it again. One of the more appealing aspects of being outside was the multitude of wildlife and insects that we encountered, such as this roly-poly that looked cool, or this common snapping turtle that we moved to Natives Pond, or the little competition that we had amongst each other who had the biggest banana spider in their area. The one in the upper right-hand corner is actually only the second runner-up or the third place, so the largest I didn't have a picture. One of the other ones that I would like to know is this monarch butterfly that was still wet in the morning and it stayed on my head for about 30 minutes. And so while it dried, it was just walking around with me. Here's a general list of the tasks that were done either on a weekly or daily basis. I particularly liked being a plant barber for plants that needed some fresh cuts, as well as collecting seeds from the Pavonia lacio patella in the natives garden. Some of the more major projects were designing, spotting and planting in 2 beds, which was one in front of the Renaissance formal garden and the children's tiers. Additionally, we also weeded whole beds. Here's a couple of pictures of those tasks being done. I enjoyed working with the Anthurium gracile seeds. I tended to gravitate towards the more tedious work that required to do little things, as well as I enjoyed working with the cactus, and especially on days that they were nice and didn't leave little spines in me. And now for medicinal plants. One of the areas that I grew particularly fond of was the prehistoric garden. When finding something to research, I had realized I'd always had an interest in medicinal studies. And one day while weeding in the prehistoric garden, go figure, it clicked. Plants that were historically or traditionally used for medicine before synthetic drugs or synthetic chemicals dominated the medicine world today. So before they did so, roughly 80% of drugs were derived from plant materials. Today, that number is down to 25%. And one of the things that ensures that plants are essential to the medical field or in medicine is that chemists can't always replicate them. Additionally, there's cost factors for modern drugs. Some tend to be too expensive for people, so they resort to traditional or natural plant remedies that our ancestors knew worked, although they did not understand how or why, they just did. Here's a couple that you may have come across, such as Resprin. It comes from or can be extracted from the Rolofia plant native to India. This is typically prescribed to patients with hypertensive patients. Additionally, Dioscorea, which is a yam native to Mexico in the tropics. Dioscorea consists of many saponins that are used by chemists to create semi-synthetic hormones. Additionally, this is a photo of a fossilized ginkgo, which I found really cool. And this brings us to our first plant, the elephant yam. It is native to the tropics of East Asia. It is part of the Araceae family. Members of these family tend to contain calcium oxalate, which is toxic. But luckily, this compound can be broken down easily by heat, so once it's cooked, it's fine to eat. This is one of the things that makes it a major crop in the area, that it's edible, as well as the broad range of environmental conditions that it can survive in. Now for its medicinal uses in the native area, it's commonly used for curative, restorative, and stomach purposes, which means that it's used as a medicine to relieve fluctuants and used to promote a natural dietary organ function. In one of the studies that I focused on, not concerning concerning the digestive issues, is its CNS, the central nervous system depressant activity that its extract exhibited in Swiss albino mice. This was observed using aquaphotometer and rod apparatus. This central nervous depressant activity is compared to diazepam, which is a commonly prescribed medicine that does the same amount of suppressant activity. That specific medicine is that's a specific medicine is prescribed to patients with anxiety, seizures, and I believe depression. Next, we have the Hachicana. It is a member of the Potaceae family. It is native to the North Indian area, and it's in the pictures on the right, it shows how good it is now. It grew from the ground up. It did, go down during the freeze. So that is it. And within the past couple of weeks, this is a picture that's pretty recent. This plant is documented by the urinary, by the Bihar tribe to be used for urinary problems. And in this study that I focused on, the extract showed anti-diabetic potential in rats. It showed anti-diabetic potential. It exhibited repair to damaged pancreatic beta cells, which is pretty significant because those are one of our cells that restore or synthesize insulin and such. But yes, it exhibited to repair those, and from those rats that were diabetically induced. Next, we have the maidenhair tree. Some may know it as the ginkgo biloba tree. It is a family of the Ginkgoaceae family. It is native to Southeast China. This cultivar that we have in the prehistoric garden comes from the 1975 Saratoga Research Foundation located in California. This is considered to be the best form of its dense and symmetrical branching. The cultivar is specifically a male clone. Fun fact, female ginkgos produce smelly berries. In the research study that I chose for this one, which is pretty exciting for me, studied its extract, which will be referred to as EGB, and its contents are listed below. Its extract exhibited or showed increased cognitive function in patients with mild to moderate dementia of the Alzheimer's type. It may be due to one of the active ingredients, which is ginkgolide B, which it inhibits neurotoxicity of the beta amyloid, which in Alzheimer's is something that is produced and blocks and bundles itself in between neurons, which is one thing that stops the motor and all the decreased things that come with having Alzheimer's progression. But aside from that, the article consisted of clinical studies, a collection of clinical studies that both negative and positive. In the more positive aspect, the studies were long-term and of course exhibited a higher cognitive function. Whereas in the negative terms, those did not show the increased cognitive function, probably because of duration of time. 11 out of the 12 that were in the study only had up to a 12-week span, which was not long enough to probably see the increased cognitive function that resulted. Additionally, the studies that were negative had lower dosage amounts. In all the positive studies, they used a 240 milligram and higher dosage and multiple dosages. Lastly, we have the Elysium star anise tree. It is part of the Sisandriaceae family. It is native to China as well. In the native area, it is known as an aromatic and medicinal plant. The study that I chose focusing on this plant followed the chemistry of its essential oil. It claimed to be one of the earliest to focus on this, and it also said that the essential oil exhibited anti-nociceptive and anti-inflammatory activities. Anti-nociceptive, it means that it stops, is an act of stopping sensory neurons from a painful stimulus. And that is also said to last longer than anesthetics. Although these are only 4 of the plants that have medicinal studies or medicinal purposes, there are many within Mercer Botanical Gardens that have, such as the aloe vera, aloe zebrina, aloe ferox, and some may even refer to this plant as the village pharmacy, the neem tree. But this just goes to show the importance of many plants that could presume to have medicinal properties that are unknown, and the importance of them in the world, in the medicine world. And what I learned, although this doesn't sum up the amount of information that I will be taking away from this internship, I now know how to do proper cuttings, what poison ivy looks like, as well as the different types of weeds that we encountered while working. Here's my citation page if anyone is interested. I would like to say thank you to Mercer for this internship opportunity, as well as anyone that instilled knowledge that I will be taking from this internship. Thank you. Does anyone have any questions?
All right, well, I believe Rob is up next from the university.
It has stopped again. Can somebody with some technical skills? Thank you. I'll do it.
You got a PowerPoint?
I do.
Okay.
Just check.
Go ahead and hit the green button, please, Chair.
What? It worked before, I just pressed it.
Yeah.
Oh,
That was the slides. So you can move the slides, it just won't show the button. Okay, you're good.
all right. Hello, all. My name is Rob Schuyler. Today, I'm going to be teaching you about bananas. What bananas we have at Mercer Botanical Gardens. Certain specific breeds I'm going to touch on a little, as well as just some biotechnology advancements we've made with bananas. Alden, can you come up again? Sorry.
Just use these buttons.
Okay, should be good now. Yes. So I go to the University of Houston, as told. I'm a biotechnology major. I have lived in Texas pretty much my whole life, and I have a love for nature and genetics. This is some examples of some of what we did here in an internship. Yeah, replanting, moving stuff, cutting stuff. We did a lot of weeding and maintenance to all these plants. Identifying, we made flower beds, put in mulch and compost, as well as did many cuttings and learned how to basically use some different medias with them, which is cool, like perlite, sorghum moss, peat moss, sand, and gravel, etc. Some more examples. I learned about my major a little more. Top right, blast nucleotide search, pretty cool system. All right, so what is a banana? You might be wondering. Well, botanically speaking, a banana can be defined as a false berry, since it's a simple fruit, and develops without, sorry, develops from one flower without a true seed. And I will touch on that in a second. The origins of the banana are still truly unknown. Some estimate Indochina, Southeast Asia is probably the likely area in which they lived and originated. The plant likes hot and humid conditions and grows in over 140 countries. Surprisingly, I found out during my stay as well on Mercer Morning Club, which is a little activity we performed a few times teaching kids what an herb is. Banana is technically an herb. Well, the banana tree is. It is a monocot herb given its herbaceous and non-woody stem called a pseudostem. So, Carl Linnaeus, you all might know him as the father of taxonomy. He observed these fruits and documented them in his species planetarium. And only documented Musa sapientum, which is now known as the silk, and paradisiaca for the cooking banana. It has now been discovered that both of these are sort of hybrids of another. And through genomic analysis, there are many more species, and most of them are actually hybrids of the two diploid species, Musa acuminata and Musa bilbinsiana. All edibles today, edible bananas today, are parthenocarpic, meaning they bear fruit without fertilization of the ovules, and they pretty much seedless. The translation of that is virgin fruit in Latin. which it really makes sense. Over these years, thousands of years, banana growers have sort of selectively bred out certain characteristics in which they wanted, such as larger fruit, less seeds, etc. There is no true difference between a banana and plantain, surprisingly. These are just culinary terms. It goes a little deeper than that. I will explain. Acuminata is generally more diverse in sort of its variety, I guess, its diversity, while Balbensiana is less of that, more of a smaller group of banana. Most at Mercer are actually a hybrid, which is Paradisiaca. I touched on that a second ago, which is basically a hybrid of Musa acuminata and Balbensiana. And it's just kind of a more adapted name for it, rather than just saying Musa acuminata x Musa Balbensiana, basically. Okay. Yeah, so the Cavendish and the African plantain, we have that right there. It's the African cooking banana, actually, but it is a plantain. They're consumed worldwide and pretty much eaten by everyone. Yeah. Bananas have this weird sort of reproduction system. They reproduce through asexual vegetative reproduction, which is basically like parthenocarpy in bananas, making them sterile. So man has to basically further cultivate them by himself. They struggle to do this on their own. However, they can by taking what's known as a sucker. And these suckers originate from the bottom of the banana, known as the corm. And yeah, this vascular stem is what's known as the pseudobulb or the corm. It helps the plant live in adverse conditions. You can also grow them in tissue culture, a pretty new thing, which is cool. That involves cutting from this said corm and growing it in a lab with tissue culture. And some species as well, such as diploid and tetraploid species especially, bear some seeds and can be reproduced that way as well. So this is another example of the corm. This is a real-time photo underground. You see the white right there. This is the sucker. You see the baby banana plant, and you can see they're sprouting all over just from that little bit of corm. Here's a map I have created at my stay at Mercer. This is a map of all the bananas in the tropical garden we have. And y'all can see that on your way out if you'd like. It's right up there. There's over 22 varieties that I've found so far. 2 of them are unknown and still need further classification. So you might be wondering, there are so many types here. How can we tell them apart? And they pretty much look the same, as you will see. The answer to that is it's pretty hard to say. Bananas may have been coming from a single cultivar from 300 to 1,000. There's really no way to tell. Many scientists argue on this. They've narrowed down the varieties into subgroups, basically, which is kind of a label for the species or the cultivar. So A is for acuminata, B is for Balbensiana, T is for Australia musa, and S is for schizocarpa. And those are all types of bananas. You see by the number count of bananas, it indicates ploidy, which is basically the chromosome set number within the nucleus. And an example, AA is a diploid and AAAA is a tetraploid, 2 and 4. So the proportion of these letters represents the contribution of the hybrid species. So if you had an AAB, it would be 75% acuminata and 25% bulbensiana relatively. Speaking of, to get even deeper, two scientists, Simmons and Shepard, they created a scoring system to estimate more inheritance by characters. Basically, the more the characters on either side, the more the relation is to that species. Now I will give a quick tour. The bananas in our garden. First one of the coolest, I think, is the golden finger. This banana has been carefully bred to basically be pest and disease resistant to many things and have a higher bunch yield. It was made in tissue culture in the lab and has been bred through other hybrids to give a greater defense against Fusarium wilt and black sigatoka, which are two main diseases that decimate banana populations. Might be wondering what the FHIA means, FHIA. That is for the Honduran Foundation for Agricultural Research. It's basically a non-profit research organization that tries to develop more disease-resistant and stronger breeds of banana, along with other plants. Next is the Mona Lisa. This is bred to be, once again, more resistant to black Sitoka. It's more susceptible to the Panama disease. The one on the right, that is the lemon tasting banana or the sweetheart. And it is more wind and drought resistant. It has a slightly bigger root system underground, and that kind of enables it to stay put, stay grounded, and fight back against disease. So next we have the dwarf Cavendish. Many of you are probably familiar with this. It's likely what you think of when you see a banana. This cultivar, along with what is known as the giant cultivar Cavendish breed, are basically the most sold bananas of all time. So 99% of bananas are sold in the Cavendish cultivar, and the dwarf is the most sold internationally since it's very hard and weather resistant to the skin, very thick exocarp. Next, this is my all-time favorite. This is the Blue Java ice cream banana. It is very sweet and soft. It fruits vanilla, soft, kind of creamy bananas. It's one of the largest we have as of now, and it is more resistant to the cold and windy conditions. So you can see by its slight blue discoloration. Next, that one's right there on the left, sort of looks like a ginger. It is the Musella laziocarpa. It's also known as the Chinese dwarf golden lotus. It's in the Musella family, which is still argued if it is a genus itself or it's in the Ensete genus. And there's no such thing as Musella, basically. It's very ornamental, as you can see, very pretty and dashing. It creates kind of a golden star when it flowers, and it is very non-edible. Looks good though. And then these are some other bananas we have. I'm gonna skip through some of these. If y'all want me to come back later, just let me know. But these are some varieties. You can tell they kind of all look the same, but African rhino horns, another one we have to the right. And to the right of this, the Ingens, Musa Ingens, we don't have anymore. It's the biggest banana of all time, however. It was removed a few years back, sadly. Next is the Big Mike, and the best for last. It's also known as a Gros Michel more popularly. It was extremely popular before 1950s, 1960s. It was very sweet. It was much thicker skin and creamier than the Cavendish, which is the yellow banana you know today. Ultimately, it failed due to its susceptibility to the race 1 Panama disease. And it also had a tendency to fall over when it bared fruit. So the Panama disease is more popularly known as, and scientifically known as Fusarium wilt, which is basically a lethal fungi that feeds off of plants and blocks it from absorbing essential nutrients like water. The race 1 disease spreads extremely fast, and it has across the globe in less than a decade and continued to do so up until it pretty much decimated all major banana plantations throughout the 20th century. Yeah, so a pretty serious thing. Also, something bad about it is it can stay infested in the soil and it's practically untraceable, or it is, but it works very insidiously. And by the time you kind of notice it, it's kind of too late. From so many of these plantations being infested, fruit companies sought really any consumer-friendly banana that was really good enough, resistant enough to survive these conditions. So that is why they chose Cavendish. The Cavendish, however, is very susceptible to tropical race 4, which is another substrain of the type of fungi that lives in the soil. And no conventional methods so far have been able to stop this strain. So we turn to biotechnology. Queensland University of Technology Center for Tropical Crops and Biocommodities has basically been working on a sort of a substrain of tropical crops, especially bananas. They've been using what's known as CRISPR engineering to try and create new cultigens and edit old ones with the RGA2 gene, which is essentially a gene in ancient plants that used to help them fight disease really well. It's kind of lost its merit over the years, but field testing so far has been very successful. Electroporation. So CRISPR itself is essentially an immune system. From earlier prokaryotes, it's been engineered with its associated proteins to basically read, insert, replace, or delete certain genes from the DNA. The method of doing this is traditional electroporation, which has been used to give CRISPR access to these genes via opening channels in the cell membrane to allow its transport. So a conductive solution is made with the CRISPR components basically stirred up, heated and electrified, and the cells, the wall and the membrane itself especially open the pores so that the genetic material can pass. This has to do with the negative charge of the membrane and stuff. Won't get into that, but anyways, banana biotechnology, very hopeful industry. It's coming up. This method is, electroporation is one of the best to introduce CRISPR into cells. There's also what's known as Agrobacterium tumefaciens, which is another good example of a viable technique these biotechnologists use to insert their desired genes in the cell. And in all, these methods hope to improve the susceptible traits by just adjusting the genes of the cell in which the trait is expressed. So all these transgenic plant methods have been proven to work in sort of smaller plants, less complicated, yes. And yeah, cells has seen they sort of coalesce. Yeah. So, thank you all for listening to me. I'm sorry I'm not that enthusiastic today, but I just wanted to thank you all for coming. Thank the Mercer Botanical Foundation itself. Thank my supervisor, Sensei, Jacob Martin, especially. He's believed in me, which is not usual for me. Yeah. Thank you. Are there any questions?
They all survived. This winter, they all came back.
That is a good question. Oh,
Mulching over there.
Oh, good, because she keeps interrupting. That's the
oh,
fifth
I'm
time,
sorry. Yes.
you know? Yeah, I bet. She is fired. She's so fired. Damn it.
That is a tough
I'm trying to
question. I'll show
do
you
the
back
thing,
to
And he
the
starts
previous
giggling.
slides. I sort of have a stage life cycle, map,
Yes.
I guess, if you will, of the banana that I'll show you. But basically, those are suckers produced from the underground corm.
All right.
I'm not exactly sure how you can identify it. I have it right here, actually. One of the unknowns. Oh, sorry. But yeah, no, I'm sorry.
Here's your quote, dear.
Oh,
My leg kept hitting my tablet. Embarrassed.
any more questions?
Yeah.
No? All right.
Sorry.
Put on the plate.
Let's see. I gotta find.
We don't have any glasses.
What's that you?
Is this? Yeah, sorry, that's Reed.
Oh my God. I was
Is this,
such a good
is
mood.
this, are you happy with this?
I think they may still be a little too.
All right.
Could be not.
By the time everybody gets their stuff.
In this park itself? Yep. Well, we do have them growing. We have them in certain spots. They really need a lot of sunlight. Oh, I'm used to engines. Yeah, we don't carry that anymore. Well. I'm not sure. Yeah. Oh yeah, forgot to mention, this is kind of a weird thing, but everything that was shaded, you look back, okay.
Okay, great. Thank you so much, Rob. And now I will talk a little bit about our interns at the Mercer Botanical Center. I thank first my amazing colleague, Linda Costanza, for all the training she provided for our interns, both on the database, at our conservation nursery, doing inventories. And now we'll see a little bit about what our interns can do. They're both Alex and Alden learned a lot about weeds this summer. It sounds to be, this is a resounding theme in these presentations, I think. But they did an amazing job with restoring our endangered species garden. And they have other skills. They even helped push our SUV out of the mud at one of our off-site research sites. So we had a lot of fun this summer. So our first intern, both interns are from Rice University, and they'll tell you all about themselves. And the first is Alex. Alex, please.
Everyone hear me all right?
Do you mind if I remove my mask while I'm presenting? All right, thank you. All right, so hello, my name is Alex, and today I wanted to talk about Cactaceae, or the cactus family. So a little bit about myself. I'm a rising sophomore at Rice University, and I'm also help maintain the residential college garden on campus. So this summer I had the opportunity to help work and maintain the Mercer Botanic Gardens, including repairing, maintaining, and building signs for that would label certain plant species throughout the garden, including those in the Daylily Lane formal garden. And my colleague and I also created a map that located and identified every single flat crinum lily in the crinum lily bed if and when a hurricane decides to blow and destroy a few things. However, we weren't only working at Mercer Botanic Gardens. We also frequently worked offsite, for example, the East End Park at Kingwood. There we were able to monitor and collect seed pods of green milkweed with the hopes of eventually restoring native populations in places like the Prairie Dawn Preserve. So the Prairie Dawn Preserve is small area of land a few miles southwest of Mercer Botanic Center. And it's important because it holds several endemic species, including Texas Prairie Dawn, its namesake, Ray Jacksonia aurea, and the Texas windmill grass. However, it also holds several invasive species, including a King Ranch bluestem, deep-rooted sedge, Brazilian verbena, Johnson grass, and it's not just restricted to plants either. We've seen pig scat and tracks. The invasive feral hogs like to stomp around on the preserve, think they own the place. And of course, there's going to be litter that blows in from the surrounding buildings and roads, and we would be in charge of cleaning that up. So back at Mercer Botanical Center, my colleague and I, we were in charge of updating and georeferencing the database of Merca specimens. So Merca, again, is the core collection at Mercer of the core collection of herbarium specimens. And throughout the summer, I was able to work on, in total, 46 families, 212 genera, 433 species, and a little over 1,100 specimens total. So a few notable families that I was able to work on include the bean family, magnolia family, the fern family, and most importantly, the cactus family. So the specific genus that I want to discuss today is the prickly pear, or Opuntia. So at the Prairie Dawn Preserve, Alden and I, we were able to collect a specimen of Opuntia humifusa, the eastern prickly pear. And as you can see, you really require the whole entourage to prepare a proper specimen. So at the cost of our hands and fingers, we're able to create a nice layout of our Opuntia humifusa. So in this picture, I sort of wanted to talk about the structure of a prickly pear cactus. In this, as you can see, there are a total of 3 pads, which are scientifically known as cladodes. And as you can see, the stems themselves are flattened and flattened stem joints. And this is the distinguishing feature of a prickly pear cactus. It distinguishes them from its close relative Cylindropuntia or the cholla cactus. And on each cladode is an arrangement of these bumps called areoles. Here's one right here. And from these areoles grow spines and these tinier spines called glochids. Now, the glochids are they're the real threats whenever you're handling a cactus. They're barbed, they're fragile. They really jump at every opportunity to stick onto your skin. It's a lot of fun. But the interesting thing to note is that since they are modified leaves, and this is most apparent when you have a young growth. For example, in the top right, you can see a picture of the young cladode, and it's a little bit hard to see due to the lighting, but each areole is covered by these fleshy growths, which most closely resemble these leaves. They eventually do fall off in place of the spines though. So as for the flower, it is quite large relative to the size of the opuntia itself, and very pretty and definitely very conspicuous to the pollinators such as this beetle. Let me point it to you right here. You see it? There you go. So the Opuntia humifusa has one of the largest native ranges of a cactus in North America. It stretches down below Texas into Mexico. However, it also goes along the East Coast up into as north as Canada. However, there's also a more native localized species, Opuntia engomanii, variety Lindheimeri. It is a prickly pear that is mostly restricted to Texas and sometimes in the surrounding areas. On the other hand, you have the Opuntia stricta species. This is a species notoriously invasive in many different parts of the world, including Australia, Kenya, even the Caribbean. And people in these places have been able to use biocontrol agents to fight back against the invasive Opuntia stricta because they are extremely resilient and they're very capable of reproducing, especially in those environments. And I also want to discuss the most commercially important cactus species, the Opuntia ficus indica, or the Indian fig prickly pear. So as you can see in the picture, they are cultivated just like any other crop for their fruits, and even their pads, they're used as a vegetable. And while they're not native to America or to the United States, they have been introduced in many other countries. It's native to Mexico. And I also wanted to talk about the collection locations of the Opuntia that we have at Mercer. Most of the species have been collected throughout and around the Houston area, with a few located around the Gulf Coast. So the Texas prickly pear is well established in Texas, but endangered species do exist. These include Opuntia arenaria, Opuntia auraspina, and Opuntia imbricata, variety Argentea. So the primary reason is usually due to habitat loss or because of Houston's urbanization. But in the case of the imbricata variety Argentea, their already narrow range is becoming more and more, or is shrinking more and more due to nosy collectors trying to have a nice cactus for their garden or their collection. So ecologically, the prickly pear is a significant food and water source in the desert environment. Their fruits especially are desired by many small rodents and birds. They tend to eat the flesh of the fruit, but they also go for the seeds. However, larger animals such as deer kind of shrug off the spines and just eat the pads themselves. And there are also birds such as the northern bobwhite quail do use the prickly pear as habitat and nest for their nests, sort of seeking protection from predators. And culturally, the prickly pear plays a very significant role, especially in Mexico. Its depictions of it can be traced back to Aztec documents such as the Codex Osuna, in which the primary capital city, Tenochtitlan, is represented with an opuntia cactus. And this carries on over to the Mexican coat of arms, which is now seen on the Mexican flag. So the Aztecs and Mayans themselves would use these opuntia cactus for spine. They use their spines as needles. They use the pads for medicinal purposes. And of course, they use food. They use them as food. But the use with the most implications would be them using dye, them using the cochineal insects to create dye. So overall, prickly pear is iconic to Mexican culture, which carries on over to Texan culture. So economically, they are edible and they are considered a major export in Mexico. And historically, they have been used as a major dye producer for the dye industry. At the time, whenever cochineal was discovered, it was one of the, if not the most strikingly red color at the time. And major nations such as England would use this dye to dye their military uniforms, and Roman Catholic cardinals would dye their garb with this red. So the cochineal insects, they're these little bugs that like to burrow into the prickly pear themselves, which natives would harvest and eventually grind up and squish into a pulp, which would get you the strikingly red color. However, they are invasive in other parts of the world, so it's not always economically beneficial, especially in Kenya, where you would have ranchers struggling to feed their livestock due to the Opuntia stricta's massive spread. So I would like to thank the Niles family, the Gibson family, Texas Natural Master Naturalists, and the Native Plant Society of Texas, and last but not least, the Mercer Society. And thank you for listening. I would like to open the floor to any questions.
So most populated Indian.
That would likely be
Invasive species is more common, or the
no. The Opuntia is actually native to the New World, so North America. So they're not considered invasive. I might put I can't say for sure, but I would, if I had to choose, it would probably be Opuntia ficus indica or Opuntia humifusa. Just has a gut feeling. No, the fruits are different enough, and it's one of the main ways to distinguish them. The genus itself is pretty convoluted, so resources are pretty conflicted on that end.
Any other questions? So Alden, our IT expert,
Okay. Hello, everyone. My name is Alden Sears, and today I'm happy to present to you about the creosote bush, which is also known as Laurea tridentata. So first, a little bit about myself. This will work. I was originally born in Kansas City, but grew up in Costa Rica. I lived there for 10 years, and that's where kind of my interest in biology in general was born. And then I went on this hike with my dad to the highest peak in Costa Rica. You can see us holding the Costa Rican flag there at the peak. And I saw stuff like this all the way on the left. And I just looked at interactions like this between different organisms and wondered, what is going on here? So that really narrowed my interest into ecology, which is generally the interaction between living organisms and their environment. And now I'm studying ecology and evolutionary biology at Rice University. And this next year, I will be a rising senior doing my senior thesis work, which involves the creosote bush. So first, I'd like to go through a little bit of what I did at the Mercer Botanic Center. And that involves, as Alex was describing earlier, more herbarium databasing. And he already explained a little bit of what we did there, so I'll just run through my numbers. I was able to proof and georeference 45 families, 191 genera, and 414 species, which rounded up to about 900 to 1,000 specimens. And here's a table of some of the most significant families that I was able to proof and georeference. You can see the largest by far was the grasses, followed by families like the snapdragon family, the myrtles, and then my family, the creosote family. So something else we did in the Mercer Botanic Gardens was a bunch of weeding in the endangered species garden. And this grooming basically allowed us to display the actual endangered species that are there rather than all the weeds and saplings that are always growing up against them. And then we also helped pot and propagate Texas natives at the Northside Nursery with Linda and take care of the nursery area in general, which involved, again, doing just weeding around the tables, as you can see in that middle picture, and excluding basically pests and herbivores that were trying to eat the plants. And then we helped pull plants out of the greenhouses up at Northside for a Father's Day plant sale. We also were able to weed the front pollinator beds at the Mercer Botanic Center, which are basically just pollinator display beds, and assemble and set up 2 portable work tables, both behind the center and at the Prairie Dawn Preserve, which is the offsite place Alex was talking about. And then we went to an offsite location in Montgomery County with Texas Parks and Wildlife to assist with a survey of transplants from 2020 of this plant called Hibiscus daisy calyx, also known as the Necus River rose mallow, which is endangered and native to Texas. So now I'd like to talk about my more specific project, which is on the family Zygophyllaceae. And this is commonly known as the creosote bush family. It's a smaller family that has only 22 genera and around 280 species, depending on nomenclature. It's found mainly in New and Old World tropical and arid regions. And here in the US, we only have 6 genera, of which we have 4 genera and specimens at the Mercer Botanic Center. And just a little bit of significance for this family. One genus, Guaiacum, is particularly known for its hard wood and medicinal resin. It's one of the hardest woods in Northern America. And Laurea is an ecologically significant genus in North and South American arid regions, which is partly why I'm doing my project on it. So at Mercer's herbarium, we have 9 genera in total, and 4 of those are our North American genera. We have 15 species in those genera and 61 specimens. And 3 of these genera and 4 of the species in those genera were introduced, so non-native to their area of collection in the United States, including Guaiacum sanctum, Plectrocarpa arborea, and Tribulus terrestris and Tribulus cystoides. And 2 genera were only collected in Mexico, including Morcillia and Fiscinoa. So here I have some range maps for those genera. All 5 of these are present in the United States, as you can see, but only 4, the green ones in particular, are native to the United States, whereas Tribulus is an entirely introduced species. So it doesn't belong here. And as you're about to see, it's super annoying. So I want to cover 2 significant species besides Laurea tridentata, which I'm going to talk a lot about later, that are from this family. So one of them is Calstremia parviflora, and it's got the largest North American range of the Calstremia species or genus. And it is a weed, but it's got very pretty flowers. It grows mostly in dry soils near roads, so it doesn't cause a lot of problems. Whereas Tribulus terrestris and the closely related species Tribulus cystoides are both noxious weeds that are widespread all over the United States, as you saw in that previous range map. And they're toxic to livestock and have spiny burrs that can damage wheels, feet, and paws or hooves. So Tribulus terrestris is really annoying. And there's some specimens up here if you want to look at the spiny burrs on the plants, as well as the other plants that Alex and I brought today. So here's a map that I made of our Calstremia species, or specimens rather, at Mercer. And as you can see, most of them are the red Calstremia parviflora around Texas and Oklahoma, but you've also got some other species closer to Houston and further out from Texas, mostly collected in southwestern United States, as is consistent with the maps from before. And then here are where we have collected Tribulus specimens from Mercer. Across the United States, it's mostly Tribulus terrestris, but as you can see, there was 1 specimen of Tribulus cystoides that was collected in Florida. And now, finally, I'd like to move on to the plant of my heart, Laurea tridentata. This is a woody shrub that is dominant in dry and desert regions like the Chihuahuan Desert, the Mojave Desert, and the Sonoran Desert. It only lives in North America, which involves southwestern United States and northern Mexico. And it is closely related to a South American species called Laurea divaricata, which is also called creosote. And they both also have the common name gobernadora in Spanish communities, which means governess. And that refers to the way that they're basically a successional plant that comes into desert grasslands or other areas and dominates over other shrubs and grasses and just establishes communities that are almost purely Laurea tridentata. So some other characteristics include it has opposite leaves on its stems, and in each one of these leaves, it has two fused leaflets with a falcate shape, which is kind of sickle shaped, as you see in the picture there. It has 5-petal yellow flowers and fuzzy whitish haired globose fruit capsules. And whenever it rains in the desert, it has really strongly scented leaves, although this is rare and I didn't really get to experience it. And it's eaten only really by small mammals and reptiles. So it doesn't really have herbivores that are causing it trouble. And then some fun facts include that Lurea tridentata has some clonal individuals that are estimated to be around 10,000 or more years old. It's used medicinally in some cultures, especially by indigenous peoples and in Mexico. And it is extremely vulnerable to desert fires, which also means that as that is one of its biggest natural enemies to expansion, basically, because we're stopping a lot of forest fires, it is encroaching on grasslands. So here's a couple images of the specimens that we have at Mercer. Again, these are up here on the table. And one was previously labeled as Lurea divaricata, which, as you'll recall, is the South American species that I discussed. But it was collected in Big Bend National Park here in Texas, which makes no sense. So I looked up some papers. I looked at the morphology, just meaning basically the physical structure of stuff like the leaves, and morphologically determined it to be Lurea tridentata. And here are where those specimens were collected. Again, mostly in Southwestern United States and Mexico. Although we have, for some reason, a gap in Arizona. I guess no one wants to go to Arizona. So something really cool about Lurea tridentata, especially for an ecologist nerd like me, is that it has 3 distinct cytotypes. And for those of you who don't know what that is, a cytotype is a characteristic regarding the genetic material of a cell, or rather a whole organism. And Lurea tridentata, in this case, has diploid, tetraploid, and hexaploid populations, which have respectively 2, 4, and 6 copies of their entire genome in each cell. And between these populations, there are geographical and genetic differences, which have already begun to lead to some degree of speciation. So they're all regarded still as the same species, but in the future, however many hundreds or thousands of years in the future, they may be considered different species. This paper in the middle that you see here talks about how, because of differences between the physical characteristics of the plants, such as when they produce flowers, there's a degree of assortative mating between cytotypes, which means basically that if you're part of one cytotype, you only tend to mate with plants of the same cytotype rather than of another, which is just a barrier to gene flow that can cause speciation. And then the other paper on the bottom talks about how that then can shift the biogeography, which is basically just where things live, to put it very simply, of associated species. So then I'd like to talk a little bit about the medicinal uses that I talked about earlier. And Lurea tridentata has been found to have antibiotic and antiviral properties outside the body, but also associated with different extracts, as you'll see here on the left. And then it has the traditional value, again, in Mexico and to indigenous peoples. Its tea and other extracts have been used to treat a wide variety of issues. You can see them in the middle. I'm not going to list them off, but if you really are curious, you can look at my references. In this paper, Arteaga et al. Has just like a 100 to 200 item long list of the things that's been used to treat. And I don't know what to believe, honestly. And then It is banned in the United States by the FDA because of its potential to cause liver damage or toxicity to other cells, such as in the brain, because of that antioxidant that inhibits bacterial growth, which is, I'm not going to try to pronounce it because I failed last time I tried to, all the way there on the right. And then finally, I'd like to wrap up by just talking a little bit about my senior thesis research at Rice about this plant. So the context for this research is that in areas like the Chihuahuan Desert, which is where I was in June and May, there are basically ecotones of places where grasslands and shrublands meet. And the creosote bush is a shrub that is encroaching on grasslands, basically winning over some of the territory that the grass currently lives in areas like the Sevieta National Wildlife Refuge. And that's where you saw one of my pictures from earlier. So the question we want to know is whether the advancement or encroachment of this bush, the creosote, is pushed by highly reproductive individuals in high density areas where it's already extremely common, or pulled further along by plants that are reproducing a lot of seeds and new plants in areas where it's rare. And that can have a significance basically to the genetic structure of the community, because a pulled wave will allow something called gene flow, which is just basically the random loss of some genes to cause the population to lose genetic diversity as it advances. And that's what you see basically in the colors on those two different graphs. So one way that we're trying to look at this is by looking at pollinators. So we went out to a desert in New Mexico and we set up a bunch of traps, like these yellow sticky traps and blue bee bowls all the way on the right. We walked 500-meter transects. We counted plants, we measured density, we measured the number of flowers on each plant, as well as capturing our specimens and placing them in bags, which I'm going to be checking out next semester. It's going to take a long time because I don't know insects. But we want to see basically where are pollinators the most common and where are they the most diverse, because that will have significance to whether individuals at low density areas versus at high density areas are reproducing the most. And that's just one piece of the puzzle kind of. So here are my references. And I also want to thank everyone who helped along the way, including Anita and Linda, as well as the Knowles family, the Gibson family, the Native Plant Society of Texas, and the Mercer Society for their generosity in this internship. Thank you. Has anyone got any questions? Yeah.
Why you was banned from the FDA?
Oh yeah, great question. So the primary reason it was banned by the FDA was because of this acid. I'm going to try to pronounce it now. Nordohydroguaretic acid. And it's an antioxidant, which means it inhibits the growth of some bacteria, but it can also, in high concentrations, cause damage to human cells. And basically, when you have a use that isn't regulated, like people just making all these extracts at home or buying them from safe places, they could overdose on that acid, and that can cause a lot of damage to the cells. So that's basically why the FDA has banned it. Yeah. Yeah.
So the tribulus, the pumpkin everywhere.
Yeah.
How did it get here? And why was it?
I am not entirely certain how it got here, but my guess and my intuition, just from what I read, is by livestock, because that's how often it is carried along. It gets stuck in something's paw or its hoof or whatever, and then deposited later on when it kind of crumples up. So sorry, that was how was it introduced? You had another question. No.
It's everywhere here.
Yeah.
Doesn't have any natural.
Yeah, not really.
Possible that was introduced as a weed in some sort of fodder or something brought in from outside the United States. That's the case.
Anyone else? All right.
Thank you all. Then.
Thank you.
And now I'd like Anita Schiller from our mosquito biocontrol initiative program, Harrison County Precinct 4. Talk a little bit about Joseph Johnson, her intern.
Howdy. Thank you, Anita, for that beautiful introduction, but I will turn things over to our intern, Joseph, very quickly. For those of you whose faces I'm not familiar with or that don't know me, founder director of a really small program which has undergone significant branding changes over the last 10 years. We were first called MAP, then DRAC, then DRAC MAP, and now we're Biological Control Initiative, or shortened to BCI. While our names changed, our mission hasn't. We're still interested in finding local occurring native biological control organisms that have a native status that we can use indiscriminately, produce them in the lab, and release them to help us fight against pest mosquitoes. Joseph came to us a little late in the internship program at no fault to himself. But when he did arrive, he jumped right in. We have a beautiful team that took him under their wings and showed him the ropes. He helped out a lot with everyday tasks, which are always new to anybody that joins our team. But he's also gotten to work on some of his own projects. And without further ado, I'd like to invite him up here that he can tell you all about all those projects and those tasks.
I may need assistance with the shared screen. I'm not sure how to do it properly.
Do you have a PowerPoint?
Yes, it's on the top right corner.
This is your presenter.
How do you advance it?
Oh,
This is it.
yeah. You gotta click on the app again because it forgets where you are. And now it should go.
Okay, thank you. Hello, everyone. My name is Joseph Johnson, and I interned this summer at the Harris County Precinct for Biological Control Initiative, which we call BCI. A little about me is I have my bachelor's degree in plant and soil science from Sam Houston State University. And currently I'm attending Lone Star College for the biotech program to get my advanced technical certificate. And my hobbies include herping, which is essentially birding, but for snakes and other reptiles and amphibians. And I've been doing that for about 20 years. And I recently got into photography as well. Been taking a lot of pictures of snakes and bugs and anything interesting in the woods. I'm also very interested in genetics, especially plant genetics and microscopy. I purchased my own microscope and started looking at pond water for the last couple of years, just in my spare time for fun. And that's kind of what brought me to BCI, is all of those hobbies, oddly enough, combine perfectly to suit the needs of BCI. BCI's mission statement is to identify, evaluate, develop, research, and find naturally occurring native organisms to use as effective control tools for integrative mosquito management programs, which target medically important mosquitoes, such as the yellow fever mosquito and several other introduced species. And biological control in itself is a facet of integrated pest management. To perform, the organisms themselves are referred to as biological control agents. And currently BCI is working with these 4 species here, as well as a few others are up and coming. And I'll talk about each one of them in just a moment. And my role at BCI this summer has had three different sections: insectary work, field studies, and project work. And in the insectary, we have our BCA colonies. Toxorhynchides rutilis is one of our major players. It's known as the mosquito assassin. It is a mosquito that lays its eggs in the similar areas to the other mosquitoes, and their larvae are predatory. Romanomyrmex culissivorax is a parasitoid nematode which infects the larval stages of the mosquitoes. And Amlongicetus is a predatory copepod, and we have that species among many others that we've collected to test against mosquitoes. We also, in addition to those, have prey and host cultures such as Aedes aegypti, which are the yellow fever mosquito, because we need to grow their enemies to see how they perform against them. And darrow worms are a freshwater segmented worm. They are quite small, about 2 centimeters or so whenever they're adults. And we feed those to the larvae of the tox mosquitoes. And then Panagrillis are microworms, which are used to feed the very small stages of the mosquito development. And then Paramecium are used to feed the copepods, as well as a few other cultures that we have. And here we have pictured the tox mosquito preying on Aedes aegypti. And they're not only predatory, but also cannibalistic. And as you can see on the left here, we have every single one of them is reared individually in its own small well. And then the Romanomyrmex culissivorax, the parasitoid nematode. Their larva, their hatchling stage is microscopic, and they infect the smaller stages of the larva of mosquitoes. And you can see one here that has nearly completed its life cycle, or this stage of its life, by eating most of the organs within the mosquito larva, but not enough to actually kill it. And then it comes to this stage right here and coils up. And then once it does emerge, it actually kind of blows the mosquito larva apart and kills it. And then once it's in that adult stage, then they will mate and lay eggs in the water to start the next cycle. I've also participated in field studies, such as population dynamic studies, which is finding out which mosquitoes we have around and whether or not an area needs to be inoculated with BCAs, as well as impact surveys to see the inoculations that we have made, how they're performing. And pictured here is me setting up a mosquito trap at Kissing Tree Park, where we are using pheromones and CO2 to attract pest mosquitoes that are searching for a host, and your breath is one of the ways that they find you. And so this attracts them, and then they get sucked into a bag that we put into a freezer so that we can identify them to see what species we have, and we do this weekly. And then in the Houston Arboretum and Nature Center, as well as the Harris County Precinct 4 Spring Creek Greenway Trail, we have inoculated with the MERMs, which is a shorthand for the Romanomyrmex parasitoid nematodes. And we return weekly to collect samples from the pond waters there to see if the mosquito larvae are indeed infected, or to reapply as needed. We also perform predatory surveys. So this was at the Telge Retention Pond. And essentially we go out to see what other predators are actively present in the area to see if it's worth adding a BCA. And also at the Telge Retention Pond is Utricularia gibba, which is also known as the horned bladderwort. And that is a carnivorous plant that is aquatic and has no roots. So it just free floats in masses and mats. And the traps actually have evolved, it has evolved traps to replace the roots so that it can trap insects and smaller aquatic organisms that it digests. So once they trigger the hairs, it's very similar to a reverse Venus flytrap, if you will. It has negative pressure, and once they touch it, it just kind of envelops them and dissolves them. And they are large enough to be predaceous on the mosquitoes of interest. And we have field collected some samples and are for the wild sourcing to put in our production line. And Mercer Botanical Gardens is actually assisting in our construction of a shade structure because sunlight here in Texas is just a bit too harsh for them. They're typically found underneath other vegetation in the ponds. And then we also have the copepods, which are, they were once referred to as zooplankton, very small, nearly microscopic crustaceans. They're very carnivorous, most species at least, and they're also very prolific. So once we get them into a colony, they multiply rather quickly. And they're of use as a BCA because they are large enough and ravenous enough to take on mosquito larvae, sometimes larger than themselves. And here's how that one played out. And then while they're easy to find, they're not necessarily easy to identify. And so we have taken many different samples of copepods from around the various areas that we've been active and set up individual females into monocultures and letting them grow for a while. And then we take a gravid female and, let me start over. We designed a preliminary predation assay in which we take a single gravid female from our individual colonies and add them to a well with 50 freshly hatched Aedes larvae, and then give them 48 hours, as well as there's also a control pod where there are no copepods present. And after 48 hours, we count how many were remaining and how many have been eaten, presumably. And Immulangicetus is with the red star above it. That one is the one that we know the species for sure. And it has a pretty good track record of eating mosquito larvae. And we thus far have 3 that are doing much better, nearly 100% predation rate. And the next steps are to find out exactly what they are, in which we've implemented DNA barcoding, which we can identify at the species level. And the process involves DNA extraction. So we select a few copepods and extract the DNA from them, and then use polymerase chain reaction or PCR to amplify that DNA. And then we send it off for sequencing. And once it's been sequenced, we compare that against published data from other scientists and what's available to us, so we can narrow them down as much as possible. And our primary focus in the last round of DNA barcoding in which I participated were the predatory copepods, of course, which are a BCA. We also did biting midges, which are another potential pest, target pest for us to begin inoculating for. And chironomids, which are an insect that lay their eggs in water and have a larval stage that are also useful as a potential insect prey. And the darrow worms, which are used to feed those mosquito larvae in their larger stages. Now I'd like to thank Commissioner Jack Hagel. Without him, BCI would not exist. Mercer Botanical Gardens and the education team for inviting me, as well as Tyler, Brandon, and Colleen, especially for helping with the shade structure and helping us get all of those things together. And the Houston Arboretum and Nature Center for allowing us to conduct our field studies there. And Dr. Daniel Kiner at Lone Star, who introduced me to the BCI team, and of course the team themselves. Thank you. Any questions?
Are the predators, how species specific are they?
Many of them are
They attack
generalists.
other insect larvae like dragonflies, mayflies, anything in the water?
Many of the BCAs that we have are generalists, and they will eat anything that they come by. It's like the Toxorhynchides, they won't distinguish between their own kind or another mosquito larva of a different species, or just anything that they can fit in their mouths really is what they'll eat. And the bladderworts are very similar. Whatever swims by and happens to brush against those hairs gets enveloped if it's small enough. And then the Romanomyrmex, they're also known as the Culex destroyer. They're much more targeted. They prefer the Culex mosquitoes, which are a lot of the pest mosquitoes are in the Culicivorax, no, Culicine. I'm still learning a lot. Yes, ma'am.
If you have a like little pond at home, and so you're encouraging all these creatures to be there, is there a problem if you add chlorinated water?
Yes. Chlorine, in essence, is going to pretty much destroy most of the bacterial and other microbial growth, which in turn is going to prevent a lot of the plants from taking hold, and less oxygen is going to be a lot tougher living for any invertebrates that make their way into the water system. The chlorine is definitely detrimental. Yes.
Is there some kind of difference between female and male copepods, or why did you use specifically female copepods in that experiment?
They do exhibit sexual dimorphism. The females are much larger and they're more easy to identify. Once they've reached an adulthood, they usually have an egg sac on both sides that are very obvious, and their antenna curl a certain way, and the males are much smaller. And that's why we use the females, because they're much bigger and much more, they need a lot more energy, so they're going to be feeding a lot more vigorously. Yes, ma'am, you had a question?
Is this plant, can it thrive in Houston, and it has to do this in an organic way of mosquito control?
Yes, and yes. They, the Utricularia gibbia that we were using is actually native to Texas and well, most of the Southeast United States. And there are several other species that are native to the US and others that are also native to like Australia. So I believe most places in the world have Utricularia species or subspecies that would be useful for an organic method of controlling pests in a small contained environment.
Maybe like a small pond, something like that. Would it float?
Initially, it does float, but it doesn't have roots. It does have a method of attaching to the soil after a certain amount of time and once it's gotten large enough, but it's not required by the plant. It's pretty much happy either way, whether it's free floating or attached.
Would it be eaten by fish or turtles?
Not necessarily turtles, I don't believe. Some fish may pick at it, but I believe snails are one of the biggest problems.
Is there a retail source for the?
There are places online that you can buy Utricularia from certain hobbyists that do carry them. It's more the specialty plant places that you'd find that.
So we're ending up at about the right point. All right. Okay, so I just wanted to ask of the audience, how many of you out there are volunteers for Mercer Botanical Gardens? All right. Thank you so much for coming. Of course, Carson has been volunteering for many years, and we really appreciate our volunteers. And how many of you are family of our interns that presented today? All right, all you family members, thank you so much for coming. Thanks for your guidance and support for all these wonderful students that we have out there. And how many of you out there are former staff of Precinct 4? All right, I knew 2 of you out there. Thank you for coming and showing your support. And I wanted to extend my thanks to our education department and our volunteer coordinator for Mercer for coming out and helping set up today. And finally, we thank our interns for their incredible level of excellence and devotion to their projects. and their fearless completion of all their tasks at Mercer. Thanks also to the Biological Control Initiative for coming out today. Impressed, how many of you came out today to support Joseph? All right. Oh, good. All right. So we thank our interns, their professors, families, friends, the staff, our Mercer Society, the Native Plant Society of Texas, Houston chapter, Heartwood, of course, always, Timber Lane Utility District, and all of our many partners and our growing Mercer volunteer family for joining us tonight. So thank you for your amazing support. Drive home safely, stay safe, and I hope to see you all out at Mercer again soon. So thank you so much. Right. We have some very nice comments from our chat. If you guys want, I'll read some of our chat and see if you can answer some of these questions. Thank you, Teresa, for saying you enjoy volunteering at Mercer. And Ruth says, very impressive speakers and projects. Sarah, thank you. Yes, they did a fantastic job. Sarah Freeman, thank you. Excellent presentations. And I never knew there were so many varieties of bananas. All right. Thank you so much, and good night to everybody.